PAMPs, which are pathogen-associated molecular patterns. These are things that are on the sort of membrane of pathogens or foreign molecules that are directly detected by PRRs or pathogen recognition receptors. So there are three main types of PRRs. There are soluble PRRs, which an example is mannose binding lectin, which allows for opsonization. So these recognise non-human carbohydrates on the cell membrane, and they then activate the lectin pathway. And then there are cell membrane receptors. There are two types which can be which can cause phagocytosis or a signalling. So there can be direct PAMP recognition. This is when there's binding to the carbohydrates, polymers or lipoproteins on the surface. There can be opsonin binding, which is when a complement receptor binds to C fragments, which I'll talk about later. And then FC receptors, which we'll talk about later, bind to opsonizing antibodies and cause phagocytosis. Phagocytosis can be done by neutrophils, monocytes, macrophages, dendritic cells, and they display different pathogen recognition receptors or opsonin receptors, which then result in phagocytosis. The other type, which is signaling PRRs, an example being toll-like receptors, these detect carbohydrates and lipids that are not usually present in healthy human cells, and they then increase the release of increased T1 interferons.